we are going over muscle contractions and the physiology that goes with it. So first of all, we're going to look from a distance at a neuron that is, for example, originating out of the spinal cord and reaches out with its axon and branches with its axon terminals to eventually have one of its synaptic end bulbs synapsing with a muscle fiber. So just imagine that this right here is the muscle fiber, and the word muscle fiber just simply means muscle cell, but we do call them fibers also because they are long and skinny. They are multinucleated, very long cells. So these are the nuclei that I'm drawing here, and they can be feet in length. And this axon also can be feet in length. So in general, both of these cells actually are very interesting in the sense that they can both end up being very long and skinny, basically. But the idea is that this neuron will send an action potential which just means that this is an electrical signal, an electrical biological signal that these cells are able to generate, which moves down this wire that we call an axon. And eventually the signal gets to these synaptic end bulbs, which are these right here, these bubble-looking ends of the neuron. And at this point, the signal now will end up generating a neurotransmitter to be exocytosed from that synaptic end bulb. And the neurotransmitter that we're going to use for this particular physiology is called acetylcholine, or sometimes we'll just shorten it as ACH. And that's going to be this chemical messenger that basically diffuses from one side of this space to the other side. So this space right here is called a synapse, and the connection between the muscle and the neuron is known as a neuromuscular junction. So this connection with the synaptic end bulb and the membrane of the muscle fiber is known as a neuromuscular junction, which of course makes sense because you've got the neuron and the muscle that are communicating in this space right here. So the next thing that's going to happen is that the signal, this electrical signal, this what we call this action potential, which is the biological electricity, which is, by the way, a propagation of an electrical signal. Propagation means a movement using ions. And ions, you must remember, are charged atoms. And specifically, what we're going to be using is sodium as well as potassium. And it's sodium with a with a plus sign on it. It's a positively charged sodium and a positively charged potassium. These are the ions that will be moved in and out of these membranes as the signal gets moved downwards. Basically, it's just going to be this exchange of these ions back and forth. And we're going to wait until we get to the nervous system to really see this in detail. So for now, just know that the way that the signal moves is that sodium goes in to the membrane and next potassium flows out. And this just keeps moving as a domino effect where sodium once again goes in and potassium goes out. So that's all you have to know for now. And don't worry if this is a bit confusing to you at the moment. The nervous system chapter goes into more detail here. But this keeps going until basically we get to the synaptic end bulb. And at this point, what we want to do is we want to magnify this image and see exactly what's happening in this space here. So let's go ahead and zoom into this right here on a new page. So we're going to draw the same structure, but just that synaptic end bulb, and we're going to make it larger. So here's the synaptic end bulb. And on the other side, you are going to have the membrane of the muscle fiber. And this membrane is also known as a sarcolemma. Now, I'm drawing some gaps here on purpose because what we're going to find is that on the membrane, you're going to have these protein receptors for the neurotransmitter. And they look like this. And so basically, the neurotransmitter is going to be found stored in these balloons known as vesicles in that synaptic end bulb. And they're stored there until they're given the signal to exocytose the neurotransmitter. So the vesicle is the balloon, and these dots that I'm drawing is supposed to be the neurotransmitter. And let's not forget that the neurotransmitter is acetylcholine, or sometimes we just say ACH for short. So what's going to happen is that if we remember, we've got this sodium that goes in, followed by potassium that goes out. And this keeps going as a domino effect, where these keep switching off. Sodium goes in and potassium goes out. But at the synaptic end bulb, we have a different ion that actually goes in. And so that's an important thing to note, and can be a very typical test question to make sure that you know this, is that it is calcium, which is also an ion. So it also has a charge, but in this case it has two pluses, so it's double plus calcium. And it is calcium that will be the next in line. So you should think about it this way. Sodium goes in, potassium goes out. Sodium goes in, potassium goes out. But then once we get to the synaptic end bulb, it is calcium that simply goes in. But when calcium goes in, this is going to cause the release of the acetylcholine from these vesicles. And so now what you need to think about is that whereas these ions were electrical, right? Electrical signals, right? In other words, it is a charge that is propagating down this axon terminal. It's a charge signal, so it's known as an electrical signal. Now, once acetylcholine gets exocytosed from the synaptic end bulb and goes into the synapse via exocytosis, right? Now this right here is known as a chemical signal. So acetylcholine does not have a charge on it, right? So it's just a chemical that now has to go and find a receptor. And that's what this one is right here. And so what it's going to do, it's going to find a binding site on each side of the receptor. There's going to be one on one side and one on the other side. And this receptor is also a gated channel, meaning it's like a gate that has to open and then close. And so right here it's closed, but we can draw it also in the open configuration. So this would be open with a space allowing for something to come in here. And it's open when basically two acetylcholines are attached to the receptor, one on each side basically. That's what needs to happen. So if none are attached, it stays closed. If only one is attached, it stays closed. But if two attach, these gates open up. So let's get some of these open with acetylcholine attached. Now I'm drawing it a bit larger here, but it's supposed to be the same as these specks that I drew elsewhere, just to make it a little bit easier to see, right? And so this is the same stuff that was being stored inside these balloons, right? You've got these balloons, the vesicles, and within the vesicles you've stored this neurotransmitter acetylcholine, and here it diffuses to the opposite side of the synapse. So we want to get them all open eventually. And so the question would be, well, once they're open, now what happens? So they're open for something. And the thing to keep in mind is that acetylcholine never actually enters the muscle fiber cell, right? So just imagine that everything on this side, okay, everything here, right, inside this space is muscle fiber cytoplasm, for example. And we're going to add to this and build it and draw more stuff in here in the next picture, right? So for right now, we can just keep it empty. But just understand that the signal has to now go into this muscle fiber, right? And so the question is, how does it go? And the answer is that we go back to the electrical signal. So think about how that's going to look like before I give you the answer. So we've got this electrical signal that's sodium and potassium, and that's the standard of electrical signals basically, sodium and potassium. Then it's calcium just at the very end. And then it goes to a chemical signal, which is this neurotransmitter that hits the receptors and opens up these gates, right? They were closed, and now they start opening. And now they have to open for something. And the answer is that it goes back to electrical, which means it's now going to be sodium that enters, right? So sodium was there all along, I just didn't draw it in. These ions, these known as electrolytes, right? They're called electrolytes because, after all, they're creating these electrical signals for us. They're throughout this fluid, right? So these body fluids are just filled with these electrolytes everywhere. So they were there all along, but now when these gates open, sodium starts to rush in to the muscle fiber. So sodium was inside the synapse, just floating around, waiting for these gates to open, and then it went from a higher concentration outside to a lower concentration inside. Now, because we're going to bring all this sodium in, this cell, which was resting, known as the resting membrane potential, it was resting with a negative charge. And again, don't worry too much about why this is the case or how this works. The nervous system chapter will go into more detail on this, but just know that this muscle fiber was resting in a very negative state, and it was waiting to get activated, right? And then once all these positively charged ions rush in, now it changes and it goes to a positive charge. And we call this from negative to positive, we call it a depolarization, which is the beginning of this electrical signal now being part of this muscle fiber. So it used to just be in the neuron here, that's where the electrical signal was. But now with acetylcholine as the chemical messenger going from the neuron to the muscle fiber, now the electrical signal now was given basically to the muscle fiber itself. And so this is going to create this effect of starting the process to basically wake up this fiber to eventually get it to contract. And so just to see where we're at right now, we have two pictures that we have. We've got this one right here as a summary, where the electrical signal starts way up here as the action potential and moves down with these ions swapping themselves. Then it goes to these terminals. Then it goes to the synaptic end bulb. And then the electrical signal turns into a chemical signal within the synapse. And then the chemical signal goes back to an electrical signal once it hits the acetylcholine receptors that are found on the sarcolemma, right? Which is another word for the cell membrane of the muscle cell. And so this is just the zoomed in view of that exchange from an electrical signal with sodium and potassium, finally calcium, to a chemical signal with acetylcholine being exocytosed, back to an electrical signal with now sodium going through these acetylcholine receptors that are found on the sarcolemma. And so now what we want to do is we want to see what happens next. There's this electrical signal now on the muscle fiber, but there's much more to the story. This is just the beginning of how the muscle fiber gets activated. So now we want to zoom back out a little bit and kind of go in between those two pictures that we drew in terms of how much we zoom. So let's go ahead and draw a synaptic end bulb. And let's draw our sarcolemma, which has a little bit of a fold here, but then straightens out a little bit. So that's our sarcolemma. And now what we want to do is we want to imagine, so the acetylcholine is happening and all that, and so you want to use the previous picture for that detail. But now what you want to imagine is that the sodium switches with the potassium again, right? And we can draw it off to the side here. So sodium goes in, potassium goes out, right? And this is going to now start moving away from this neuromuscular junction and outwards to spread out over this muscle cell membrane. So let's start moving it out to the side. There's my sodium going in, there's my potassium going out, and so on and so forth. And this just keeps on going. So now I'm just going to draw arrows going in and arrows going out. And you just have to know that it's sodium going in and potassium going out. So at this point, instead of drawing the ions over and over again, which you can do in your own notes if it helps you, but I'm just going to draw arrows at this point as the idea that you now know which ions are doing this. And it's going to spread out this way too. Right, sodium goes in, potassium goes out. Sodium goes in and potassium goes out, right? So this just keeps happening. Now, what happens to the sarcolemma? The sarcolemma ends up diving deep into the cell itself. Creating what we know as T-tubules. So this is the sarcolemma. This continues, right? This is just one part of this very long cell, right? So this sarcolemma will continue, let's just say for inches and inches, or feet and feet, however you want to think about it. But basically, it does create these tubes that go into the cell. And I would suggest you also look at your textbook images of these tubes. In fact, I do have a textbook here so that I can show you what we're looking at. To be clear on this, because obviously my pictures are not as good as the textbook images. So what we're looking at is this right here. And it's this, so this is the sarcolemma. This, in this case, it's blue, this light baby blue in this picture. And you can see that this tube is also that same blue. And the reason why it's the same blue, it's just an extension of the sarcolemma, right? And so there it goes, it dives in, and then this is the inside machinery, right, of that muscle fiber, right? And these can be very long, and so this is just one little bit of it that we're gonna learn how this bit works. And if we can learn how this part works, we can learn how it works at any point throughout the length of this cell. But I do want you to look at this T-tubule. It's T-tubule for short. It's actually a transverse tubule. That's the actual name, transverse, written out right here. Transverse meaning it transverses, or it goes through, right? And you can see how this one's kind of going through. So these are transversing or going through the cells. And then notice that in yellow, you've got this other stuff, which is known as the sarcoplasmic reticulum, right? And the sarcoplasmic reticulum is another very important organelle that is, if you remember the endoplasmic reticulum from general biology, it is a specialized endoplasmic reticulum for muscle contractions. And so what we're seeing here is how these two are right next to each other, and how these two are going to be communicating with each other. And then over here, what you've got is, in here, you've got the myofibrils, which are these cylindrical contractile structures, and these myofibrils are made up of the proteins that do the actual contractions. And then if you take a look at a slice of that myofibril, you notice that it's got these thick filament myosins and thin filament actins that are crisscrossing. And the contractile unit is known as the sarcomere, which is the space between two of these protein zigzagging Z discs on either side. So make sure you take a look at this and study all of this anatomy of the muscle fiber very well before you try to understand how all of this works. And so once again, my picture is going to be not as good as a textbook image, but it's going to be more about how all of this works. So these are my T-tubules, right? That's as good as they're going to get for my drawing. But the reason why they are important is because as this sodium and potassium switch off with each other, because this is an electrical signal, it behaves just like the electricity that is going through the wires in your own house. And so it will follow the wire. And basically the T-tubule is the wire that is now going to enter inside the cell itself, if that makes sense. It's the wire that is bending into the cell, which is going to actually bring the signal itself into the cell. So now our arrows will follow the T-tubule. And again, it's going to be sodium, potassium, sodium, potassium, sodium, potassium, right? So I'm just drawing arrows now, but if you prefer to draw in these ions every single time to help yourself understand the material, then you should. And it's going to do the same thing on this side. Arrow goes in, arrow out, right? And these are going to crisscross. It's going to be a domino effect where one leads to the other, right? So when the sodium goes in, it's going to force the potassium out. When the potassium goes out, it's going to get the sodium to come in. So it really is like a domino effect where one pushes the other one to take place. And so this is how the T-tubules bring the signal into the cell. Then within the cell, based on that textbook picture we saw, and I would suggest you have it next to you as well as you study, is that you do have the sarcoplasmic reticulum flanking it. And the parts that are touching the T-tubules are known as the terminal cisterns. Right? So this part is the terminal cistern of the sarcoplasmic reticulum. So I'm drawing my sarcoplasmic reticulum here. And I'm abbreviating it, so even though this is going to spread out throughout this whole area, I'm just going to kind of chop it off right here just to not make the picture get too complicated. So this is right here the sarcoplasmic reticulum, and again these membranes will spread out, right? And look at the textbook picture to see how this stuff just spreads out throughout. So I've cut it right here on both ends because I want to use this space inside to draw. And so what am I going to draw in here? I'm going to draw the actual sarcomere, right? And we saw before that the sarcomere is composed of thick and thin filaments. So let's go ahead and draw those in. So I'm going to go ahead and draw my thick filaments in the same pink that I drew the sarcolemma. I just don't have that many colors to draw with, so it's not the same material, but I'm just using the same colors here. So there is the thick filament, right? And this is known as myosin, right? And this thick filament will have these golf club looking proteins that are bent and have this golf club head on the ends of these curved protein structures. Now, the reason why these are shaped this way is because they're reaching out in order to grab the thin filaments, which we're about to draw in as well. So these are described oftentimes as golf club looking proteins. The actin, which is the thin filament, looks kind of like Mardi Gras beads. And so, again, my drawings are pretty simplified compared to the textbook. So have the textbook next to you to see better pictures. I did draw this one a bit, a little bit crooked. It would have been better. I'll draw it better on the other side. Kind of center it and make it go across. But the idea is that you've got these actins that are creating the thin filaments and the myosins that are creating the thick filaments. And then these actins are tied up by proteins. Again, proteins, right? Proteins is the name of the game when it comes to muscle contractions. So we're going to tie these up with proteins that are known as these Z discs and anchor them down this way. Tie them off right there, right? And these are the Z discs, and they're called Z discs, as you can tell, because it looks like they're drawing out these letter Z's that repeat and repeat. And obviously, this is very simplified. I've only drawn 2 thick filaments. I've drawn 6 thin filaments, but obviously, right, this you'd have a whole army of these that are not expanding outwards, right, but are also one on top of another, right? These are 3-dimensional myofibrils, cylindrical shaped, so there's going to be a thickness to these myofibrils. Now, what's going to happen is that, first of all, we need to understand this relationship of the T-tubule with this terminal cistern of the sarcoplasmic reticulum. Basically, the sarcoplasmic reticulum is a storage for calcium, right? So calcium is being stored within the sarcoplasmic reticulum. And this calcium, oops, drawing pound signs by mistake here. It's just double plus. This calcium that's being stored in here is going to be released once the T-tubules give the signal to the sarcoplasmic reticulum, right? So this is a wire, and the electrical signal is going down the wire. That's what these arrows are showing us. But then this electrical signal is what gives the sarcoplasmic reticulum the signal to basically release the calcium and have the calcium go to the sarcomere. Right? So now the calcium is outside the sarcoplasmic reticulum and is going to go and find something to attach to in the sarcomere that I have not drawn in right here. So we're gonna see a better picture of it in a second. So for right now, just understand that you've got the electrical signal. It's going away from the neuromuscular junction here. It's moving out, but then it follows the T-tubules in, and it gets the sarcoplasmic reticulum to release the calcium, which is then going to go to the sarcomere. And this is going to get the sarcomere to now be able to contract. And so we do have to add a few more things to this. And so the next picture will be the detail of how calcium gets this to turn on right here. So let's use the textbook again to see how they depict the next proteins that we have to talk about. So the next proteins we're going to refer to are these regulatory proteins that are found right here. One is known as troponin, which is the one that the calcium will bind to. And the one that is attached to troponin, which is this one right here, in this case this brown one, is tropomyosin. The actin is this yellow one that we have already drawn. And then here you can see how they've depicted the myosin looking like these golf clubs. And so obviously this picture is a much better drawing of these thick filaments and how they actually look. So we need to see how calcium basically affects troponin and tropomyosin to regulate the actin and to allow for the actin to have its binding sites, these myosin binding sites, exposed so that myosin can actually bind. So notice it says myosin binding site covered by tropomyosin. So this is in the off position. Okay? So having seen that picture, let's go ahead and draw our own picture, although again it's going to be rather simplified. And so mine is going to look like this. I've got my actin here. And on each actin, I've got a binding site, which I'm just depicting with these dots. These binding sites for myosin. And myosin will be trying to always attach to those dots, to those binding sites, right? It's always going to be trying, it's sticky to actin. So myosin is sticky to actin, it wants to attach. So the question is, well, how can we stop it from attaching when we don't want to contract a muscle, when we are in the off switch? And the answer is that you've got, and mine is going to be in green here, this rod-shaped tropomyosin, right, that is going to be in between, right? This is the This is the tropomyosin, this rod-shaped protein that is in between the myosin and the actin. So that myosin, even though it wants to attach to actin, it just can't if tropomyosin is in the way. But tropomyosin is regulated by another protein that's attached here known as troponin. And troponin has calcium binding sites. Again, my pictures are going to look different than the textbook, so just kind of match what I'm doing to what the textbook has. So this is the off switch, right? And just remember that the sarcoplasmic reticulum had the calcium stored, and then the calcium ended up getting thrown out of the sarcoplasmic reticulum once the electrical signal went through the T-tubules. So let's go ahead and see what happens when calcium comes in. So let's draw it again. Here is the actin. And now we're going to draw the troponin with calcium attached to it. And when calcium is attached to it, it literally takes the tropomyosin and lifts it up and removes it and moves it out of the way. And so we're going to draw the tropomyosin, in this case, up here. So tropomyosin, we can just say moved out of the way, right? And exposing binding sites for exposing binding sites for myosin, right? So remember that there were these binding sites on the actin. These little dots are supposed to represent the binding sites, or the sticky spots. You can think about them as sticky spots for myosin. And myosin now should be able to reach, okay, and make this connection right here with the actin. And it will. We just have one more thing to talk about, which is, of course, ATP. Because ATP is the energy that, of course, needs to be there as well in order to actually get the contraction to go. But at least here, what we've done is we have used the calcium to allow for the myosin and the actin to make these connections. So the next thing that happens is that with ATP hydrolysis, we are going to get this actin to move, right? And we're going to get it to move in order to get this contraction to take place. And so I'm going to draw a very simplified picture of this, and then we can look at the textbook to see the detail of this. But basically, you can think of these myosins being kind of extended out. Kind of straight when they first make their connections. But then with ATP hydrolysis, which is, right, ATP goes to ADP plus PI. Now we call it ATP hydrolysis. And you have to remember from general biology that the word hydro means water and lysis means to cut. So we're going to use water to cut this bond, right? ATP is an adenosine with 3 phosphates, right? And the energy is right here in this bond of that third and second phosphate. And so if water can come in to cut this, then we end up getting adenosine diphosphate, right? 2 P's, and then this PI is removed, but this energy, right? This energy came out of this. And what's going to happen is that the energy that comes out of this breaking of this bond between the second and the third is going to get these myosins to bend, right? And so then your next picture should look something like this, with the myosins curved. Right? So they went from this straight, reaching, stretched out shape to a curved shape. Sorry for the the funny drawings here of the actin. The actin is the same, right? No difference there, just kind of looks funny right there the way I drew it. But the idea is that the actin, when these myosins bend with this ATP, right, with this energy hydrolysis, that's the energy that causes them to bend, right? The actin moves, right? The actin moves, it shifts. And if you go back to this picture, you want to think about basically, right? There is another structure in the sarcomere that is central. It's called the M line, which is the middle of the sarcomere. It's a protein structure, scaffolding that goes down the middle of the sarcomere, right? This is known as the M line. That basically all the actins, right? This actin gets pulled towards the M line by these myosins. These get pulled this way, so they kind of run into, like they run towards each other, okay? And when all of that happens, when this actin moves that way and this actin moves that way simultaneously, same with this and this and this and this, right? Right? They're moving towards each other, is that you get this shortening, right? So the muscle shortens, and that's why we call it a contraction, because literally this whole thing, this whole sarcomere, just scrunches up and gets really, really short during the muscle contraction. And then during the relaxation, it expands again to get to its original shape, right? So you want to think about this picture and, of course, this picture, right? All together, right? You want to be able to put all these together, right? The regulatory proteins with the calcium, the ATP hydrolysis, changing this shape of the myosin, and then looking at it as this whole entire sarcomere contracting or getting shorter. So if we look at the textbook picture, we get a lot more detail about this ATP hydrolysis. And that's this picture right here. So what we notice is that if we start here, we've got this is right when the contraction ended, basically, and the myosin detached from the actin, okay? And here what they're showing us with the key here, they're saying that the calcium is this purple, right? So there's the calcium attached to the troponin, so it's already there. Tropomyosin, therefore, has already shifted out of the way. Those dots are the myosin binding sites, right? And then notice here that you've got ATP. So the adenosine triphosphate has not been hydrolyzed yet. But then if you go to the next picture, you notice that there is a hydrolysis reaction because it says ADP and P have separated, So that is the hydrolysis reaction right there. And notice what happened to the shape of this curved myosin. It extended out, right, to reach. And that's exactly what it does. It changes shape with the help of this ATP molecule. It can change shape. And now look over here in this picture, the next one. It's got this, it shows that the phosphate, right, leaves, and that's what this arrow is showing us, leaving the ADP by itself. But that's the trigger to actually get the myosin to connect to the actin. So here you finally get this cross bridge, is what this is called, this cross bridge. The connection, the stickiness is called the cross bridge, when they stick together. The cross bridge between the myosin heads and the actin right there. And then notice in the next picture, you've got the ADP that leaves. That's what this arrow is showing us. It leaves. And so it's not there anymore, but that gets the myosin to bend, right? And that's what these arrows are showing us. It's not the best picture, I have to say, but what you have to imagine is that the myosin moved, right? It moved this way. One way to see this in the picture is notice where the thick filaments and the thin filaments line up here, and you see that they're flush together. But here you notice that they're not, right? There's a gap, and that gap is the distance that the actin was shifted. And you can see now it's hanging off on this side. As you can tell there, it's kind of hanging off, whereas here it's flush on both sides, right? So that picture is not the best to show how this myosin actually moved, right? But it did happen. And then the last image is showing us that ATP comes back in to restart this thing. And what's really interesting about this is that ATP has to bind for this cross bridge to break. And so if ATP does not come back in, these will stay stuck together. But when ATP comes in, it releases the myosin from the actin, and it's going to wait there until we need to do this so-called power stroke, right? So-called power stroke all over again. So the power stroke is this, right? It's the myosin powering or moving the actins towards the M line, okay? So the next picture here is another good one, which shows us, this is on the previous page, shows us how these will, in fact, sorry if it's... So this picture, I did get it straightened out here, is showing us how, in fact, these sarcomeres do shorten. And on one side, you've got the actual microscope images, and on the other side, you've got the textbook images that are drawn out the way that all these proteins are drawn out. So what we want to see here is that you've got Z discs, right? And so in between each Z disc is going to be that sarcomere, right? So there's a sarcomere here, there's a sarcomere here, right? And then you want to notice the M lines, right? Looks like a chain of protein that's in the middle of the sarcomere there. That's the M line there. And then you do have these zones and bands that are labeled for you, right? So the H zone is the zone of just thick filaments. The I band is the band of just thin filaments. The A band is thick and thin, right, that are one on top of another. And these are just names that were given from these microscope images that early anatomists gave when they were looking at muscle tissue under the microscope. And then you can see these Z discs very pronounced, right, on these microscope images. So then what's cool about this is that it shows you what happens as these contractions take place. And so this one right here is a partially contracted muscle. And you can see that in green highlighted here versus here, the H zone has gotten smaller. Why? Because the actins are moving towards that M line. And that then with a maximally contracted muscle, you can see how much overlap you get there. So literally the actins are just sliding over each other, right? And this in fact is known, right, because of this sliding appearance, right? This whole physiology of the sarcomere getting shorter is known as the sliding filament mechanism. Because literally the idea was that these are just overlapping and sliding over each other as they contract. So now what you want to do is think about how to turn off all of this, right? And so we've talked about the details about how this turns on and how these things actually get the contraction to happen. But the question is, how do we shut this down? So one thing that you'll read about in detail in your textbook is that there are calcium pumps that are attached to the sarcoplasmic reticulum. And those calcium pumps are going to pump the calcium back into the sarcoplasmic reticulum, right? Because if there's no calcium, then the myosin cannot attach to the actin. So calcium pumps bring the calcium back in. Of course, ATP, right? If there's no ATP, we did say that ATP does get the myosins and the actins to release from each other. So if there's not enough ATP, they're going to stay more locked together, and that's going to be somewhat of an off switch. One thing you can think about is rigor mortis, which is the stiffening of a dead body. After death, the body stiffens because the muscles are all locked up. And the reason why they're all locked up is because a dead body cannot make ATP anymore. So if there's no ATP, right? If there's no ATP being made by that body anymore, because everything's shut down, so, right, no mitochondria can work and all that, then these cross bridges of the myosin and the actin will stay locked, creating what we know as rigor mortis, or the stiffening of the dead body. And then looking at this picture, we know that there is an enzyme known as acetylcholine esterase. Esterase, that's a big word, right? Acetylcholine esterase, which goes in and starts to recycle, starts to break down the acetylcholines that are attached to these receptors, starts breaking them down, and starts to recycle that material, right? Helps to recycle that material back into the synaptic end bulb so that these vesicles, it can be assembled and then put back into the vesicles to do this all over again. So acetylcholine esterase is another way to basically shut this off, right? Shut this off and get this to reset itself. Finally, one more thing that you can think of with all of this and these ions is, of course, a famous pump that you heard about in general biology, and this is known as the sodium-potassium pump, right? Na+, K+ pump. And if you don't remember what this is, very simply, it's going to take 3 sodiums and pump them out. And it's going to simultaneously pump 2 potassiums in, right? And this resets the ions that are outside and inside. Notice that if you pump the sodiums out, right, then they're found outside, and of course that's going to allow for them to go back in when we do have our electrical action potentials again. So that's a summary of all of it. You do need to take these pictures and try your best to put them all together in one big idea, so that, right, you start from one and you go to the next thing, right, and you understand this, and then, right, you go inside the cell, the muscle fiber itself, and understand the machinery found within, and then finally understanding the power stroke, and then also understanding how all of this turns off and resets itself at the very end. And so notice we're using different pictures just because it is one of the most complicated physiologies that you've had at this point, and it would be very difficult to draw this all out in one picture without it getting very messy.